Assalamu alaikum. On behalf of Ahsan Academy, let me welcome you to a presentation on gametogenesis. This part is a short presentation that will include the spermatogenesis only. So the learning objective of this part I try to cover is to define the gametogenesis and its type. Then I'll discuss the background idea on the site of spermatogenesis, steps of spermatogenesis, and events that occurs in every stages of spermatogenesis. This two histological slide you can see on the left side. This is histological section of ovary, and on the right side this is the section through the seminiferous tubule of testes. Here you see the germ cells are there which are converted to gametes. This process of conversion of germ cells into male or female gamete is known as gametogenesis. So gametogenesis may be for the female, gametogenesis may be for the male. Gametogenesis for the female is known as oogenesis, and for the male that is sperm. In this presentation today I'll try to cover the male part, means spermatogenesis. Spermatogenesis, so it is conversion of germ cells into male gamete. So it occurs in the testes. So this is a process, series of events are there that includes all the events by which the spermatogonium are transformed into spermatozoa. So spermatogonium is the immature form and spermatozoa is the final destination, the mature form. So this is the seminiferous tubule present inside the testis, seen under light microscope. So this is one seminiferous tubule that is almost complete, and these are others partially sectioned. This is the diagram showing the changing cells from outer to inside. So at the base, so outer part, close to the outer limit, this is spermatogonium. So this is the germ cell that is in mature form, divides by mitosis. So this is the germ cell who divides by mitosis and increase in number. So from spermatogonium, through a series of changes, it approaches towards the lumen and becomes spermatozoa. So this is the spermatogonium, who is the germ cell, and gradually these germ cells matures to form spermatozoa, that is the gamete, and it is male gamete. So we'll try to understand how this is transforming and what are the events that is occurring during this process. Where actually it's occurring? It is occurring in the testes. We study the testes in detail in gross anatomy. Let us have a very short background knowledge. This is the testis, and it is shown by the section with some enlargement and showing the diagrammatically the composition. So this is lobulated structure. This is one lobule containing very convoluted tubules, not one, multiple tubules are there, and each tubule is known as seminiferous tubule. So one tubule is shown here. It is blind ended, and you see the two end fuses together here, and so the tube contains germ cells who ultimately form gamete inside the lumen, and so that passes towards this side and ultimately coming out. So seminiferous tubule is the factory where actually spermatozoa is produced, and this is produced from one type of immature cell who continuously proliferating to increase the number and maintaining the raw source for the spermatozoa. So at birth, what we will find, this tubules remains as a cord, filled cord. There is no lumen inside. So the cells remain dormant and it's a cord. When the male attain the puberty, because of the hormone, there will be canalization of this and the process will start here, that is known as spermatogenesis. So canalization will be there, and from the periphery the cell will gradually mature and move towards the lumen. Here you can see the seminiferous tubules, mature one. So at reproductive age, there is immature cell, spermatogonium, dividing by the mitosis and gradually changing and becoming spermatozoon at the center. So it's a process, begins at the puberty and comprises of 4 stages. For the descriptive purpose, these are named as initial proliferation, then primary spermatocytes formation, meiosis, and finally the final differentiation. So each stage will try to identify the features and what are the characteristics actually. All are diagrammatically shown. This is the seminiferous tubules and all are diagrammatically present here. This is histological figure. You see there is connective tissue septum, connective tissue separating each seminiferous tubules who are sectioned here. So at the initial stage, the proliferation starts. So spermatogonial stem cells, they divide by mitosis, an increase in number. This cell, after some time, differentiates into type 1 spermatogonia. This having some special characteristics here. This also can divide by mitosis and increase in number, but it moves to the second layer from deeper part. So this is the actual histological section, and this one on the right upper part, this is you can see is diagrammatic figure. So actual histological figure is on the left side. You see the spermatogonium remains at the close to the base, and they continue division and then moves towards the lumen and differentiates into spermatogonial cell. These are smaller, relatively smaller, rounded cells, and they have, with the darker nucleus, almost ovoid or rounded in shape, and they continue division and differentiates into B spermatogonium. So B spermatogonium has more rounded nucleus. This might be the B, and they divide by mitosis for a few attempts, maybe 2, 3, and then differentiates into primary spermatocytes. This is primary spermatocytes accepts meiosis. So this is the primary spermatocytes level, and they start divide meiotically. So here I have discussed 2 stages. First, initial proliferation, and second is the formation of the primary spermatocytes. This primary spermatocytes is simply a differentiation process, and this is proliferation process in the initial phase. So third stage is meiosis. So meiosis starts on primary spermatocytes. This meiosis 1 makes it secondary spermatocyte. So reduction division is there. Each is with half number of chromosome. This accepts the second meiotic division, and the cell produced known as spermatids. So one primary spermatocyte will produce 4 spermatid who are haploid in nature. So this is immature still form, and this undergoes some differentiation to produce a spermatozoa. This is actually waiting for further maturation and will become a mature spermatozoa. So this kind of differentiation needs one process, and this is known as spermiogenesis, and this is the fourth stage, that is the final stage of the differentiation. Spermiogenesis is the process. So this is also having several events that transform the spermatid into spermatozoa. So spermatozoon is the singular number and spermatozoa is the plural number. This is having four stages. If you start from the left side, you will find the first stage is formation of the acrosomal cast. You see on the top there is reddish spot. This is actually forming the acrosomal cap over the nucleus. So nucleus is condensed. This will condense the nuclear materials to have a compact nucleus gradually. And third stage, there will be formation of head. Then this is the neck. This is the middle piece. This is junction is the neck, middle piece, and tail formation. And finally, shedding of the extra cytoplasm. This is shedding this cytoplasm to become a slim structure. So we have already discussed the four stages. Let us summarize those four stages. So this is the seminiferous tubule diagrammatically drawn to show the germ cells as well as supporting cell. This is supporting cell, and these are the germ cells. So gradually showing some change from bottom, and so at the bottom means deeper part there is initial proliferation, which is characterized by spermatogonial proliferation by mitosis. Then by differentiation, primary spermatocyte is formed. It is simply differentiation, and when primary spermatocyte is formed, each cell accepts meiosis, and by meiosis spermatids are formed. So number of spermatids are formed and come to the luminal surface. It is luminal surface and show gradual changes or differentiation. This is the final differentiation. This is known as spermiogenesis, and in spermiogenesis there are four stages: acrosomal cap formation, this nuclear condensation, neck, middle piece, and tail formation, and finally shedding of extra cytoplasm. So this is the spermatogenesis process, and this takes about 74 days to complete one cycle, and approximately 300 million sperms are produced daily from the testes of one adult human. Thank you very much for remaining with the presentation.